video tutorial for microbiology with Shagnik. So today we are going to discuss about another DNA sequencing method that is very interesting and also important, that we have not discussed about this DNA sequencing method earlier, that is our Maxam-Gilbert DNA sequencing method, or also known as chemical degradation method. Okay, so it was discovered by, or I should say developed by 1977 by Maxam-Gilbert, and here in case of Sanger's DNA sequencing method, we are using dideoxynucleotide, right? And the process name was chain termination. But here we are using chemical degradation method, and as the name suggests, here we are using chemicals, okay, that is commonly known as modification chemicals. Why? Because these modification chemicals, which is used to modify the bases, okay, specific bases in more specifically speaking, and these specific bases and it will help us to clear them also, okay. So that means we are using chemicals to modify the bases so that we can cut them. So what is the utility of this cutting? So by using this cutting, we will generate various radioactively labeled fragments, okay. And as we have used for different condition in our Sanger's method, here we are also going to use for different concentration of for different conditions, okay. And these 4 different condition will be separately run in a gel, and by comparing the bands we can sequence what is present, or what is that exact nucleotide sequence of the DNA, okay. That is the idea of Maxam-Gilbert DNA sequencing method. So let's enter inside this topic. So first of all, although there will be another step here, if your DNA molecule is too longer, okay, then you have to first add restriction endonuclease. That means you have to perform a restriction digestion to convert them into smaller fragments that can easily be sequenced with lower error rate with the help of Maxam-Gilbert DNA sequencing, okay. So and remember one thing, that here we are going to use a mixture of restriction endonuclease, okay. Why? I am going to discuss with you. So after converting them into smaller fragments, we will denature them to obtain the single stranded DNA. That is our officially first step, okay. So now after converting them into single stranded DNA molecule, our job will be adding a radioactively labeled P32 at the 5' end of the single stranded DNA molecule, okay. How can we add? We can add a radioactivity level P32 by using terminal T4 polynucleotide kinase, okay. This T4 polynucleotide kinase, if radioactively labeled P32 containing ATP is in your system, this T4 polynucleotide kinase will simply snatch the phosphate from that ATP and attached it in the 5' end of the single stranded DNA molecule, okay. So after attachment, you will add, or I should say divide your DNA samples into 4 reaction tubes, okay. We have divided this very successfully now. So as obvious, we are going to use 4 different conditions, right? So in the first tube, which is marked as G, we are going to use our DMS, that is the first chemical, dimethyl sulfate, okay. This dimethyl sulfate will have the ability to attack on adenine and guanine, that means our purine. Not only DMS, we can also use piperidine. Piperidine. Okay, they are also going to attack A and G. And in case of pyrimidine, we have hydrazine. Okay, so in the G, our DMS or piperidine concentration should be controlled in such a way that it will only cleave G, not A. Okay, and also our concentration should be controlled in such a way that it will introduce only one modification per DNA molecule. Okay, so as a result, we have different DNA fragment of different size. Okay, all are radioactively labeled. Okay, and of different size. Okay, so in case of A plus G, our dimethyl sulfate or piperidine concentration will be quite different from the G. Okay, so that you can also, so that it can also cut the A nucleotide. Okay, and in case of C plus T, we will add hydrazine, and C we will add hydrazine, and obviously the hydrazine concentration will be different in both the cases. Now what we have now, we have different radioactively labeled DNA fragment in 4 different test tubes. Okay, now if we add the samples of G in one test tube and samples of A plus G in one test tube, sample of T plus C in one test tube, and samples of C in another test tube, extremely sorry, that if we add samples present in G test tube in one lane, A plus G in another lane, T plus C in another lane, and C another lane, and there should be a marker also in another lane that will guide us about their molecular weight of our separated fragment. Okay, and start the gel electrophoresis, and obviously here we are using page, right? So our gel will be migrate from the top to bottom. Okay, so we will have separated fragments. Okay, and these fragments can be observed with the help of autoradiogram. Okay, now the bands we have in our autoradiogram will be read for sequencing from the bottom to the top. Why? Because there are electrodes here, the positive electrodes are present and here negative. Okay, and we all know that our DNA is negatively charged due to the phosphodiester backbone present. All our DNA samples will be migrating towards the bottom depending on their size, right? And now let's try to interpret. Let's come back here. Suppose in case of G test tube, we will have, let me take another color. Okay, one fragment and two fragment. Okay, so in case of that are the fragment mentioned here. So in case of the first one, will they move faster or relatively slower? Obviously slower, because their size is quite longer than the second one. Okay, similarly here also, right? Now remember one thing, the fragments that are in the bottom will simply indicate nucleotides that are in the 5' end. Okay, and another thing that you have to remember while you are resolving your DNA sequence with the help of this band is the bands that are present in both A and G in the same line. Okay, or that means I am willing to tell you, suppose there is a band which is present, suppose let's try to focus on that band, A plus G. This band is also present in A plus G and it is also present in G. So in that case we have to consider that here G is present. Okay, That is the idea. Although here no such kind of things are present, because this line is simply the lane. Okay, so if you find any two bands in same line, suppose there is this and this, okay, so in that case we have to consider that A plus G and G, that means here G is present. Okay, and if you have a band, suppose this one, which is not present in this one, in that case we have to consider that this band will indicate A. As you can see, this band is indicating A. Okay, similarly the band which is present in C plus T, if absent in C, as you can see, it will simply represent T. Okay, and similarly the band which is present in C plus T and also present in C will represent you the C. Okay, and in case of G and C, the band which is present obviously indicating C and G. I am just telling you about the DNA nucleotide sequence, nucleotide identification process in case of A plus G tube and T plus C tube. In that way you can deduce your DNA sequence. Okay, so let's try to deduce the DNA sequence with me. Okay, so as this is A plus G, there we have two bands. Okay, although these two bands are not present here, that means what, these two are AA. So in the 3' end we will have AA. Okay, now as you can see, let me take another color, then it will be good to you, that there is a G. Okay, so we will put here simply G, because in the A plus G portion there is no band. Now our next a one nucleotide extra containing band is this one, that is present in case of C plus T. Let me take another color to represent. So as it is present in C plus T but not in C, we can say that it is T. Okay, again let me take another color. Okay, this band is present in C but not in C plus T, so what we conclude, that it is C. Okay, again let's come here. Also I do not have any more color, that this black band is present in A plus G but not in C, not in G. So what will be our interpretation, that it is A. Okay, again let me take another color. There is a band in G but not in A plus G, so what will be our consideration, that it is G. Yes or no? Now we have a band in C plus T but not in C, that means it is T. Okay, and we have a band in C but not in C plus T. We have a band in C in the last one but not in C plus T, so what will be our consideration, that it will be C. So if you going to match this C T G A C T G A E, is it matched with the sequence that we have derived? Yes, absolutely, right? So that is how the sequencing is done with the help of Maxam-Gilbert DNA sequencing method, with the help of three chemicals, right, by simply causing degradation of specific bases. Now I think your question will be, how can you determine, Shagnik, the 5' end adenine, right? So that is the reason I have told you that if you have a very large DNA molecule, you will digest them with restriction endonuclease, where you have to use a mixture of 4 and 6 base pair restriction endonuclease cutter. So what it will give us, it will give us smaller fragment which are in overlaps. Okay, that means This funda is something like jigsaw puzzle. Okay, suppose this is a entire DNA molecule. Okay, so you have a restriction cut side here. So you have generated this fragment and considered them to sequence, right? So as a result, you have successfully sequenced all the nucleotide except the nucleotide that is present in the 5' end. Okay, now as you have used a mixture of restriction endonuclease, suppose there is another cut site present of another restriction endonuclease. Can you use this restriction endonuclease, okay, to fill up what is present in the 5' end of your DNA of interest that you are willing to sequence, just like a jigsaw puzzle method? Yes, obviously, because here your 5' end can be sequenced by this part. Okay, and let me draw it. And you have already data about what is present here, so this sequence will give you the details of the sequence that is present in that part, and this red marked portion will give you the what is present in that part. So can you use to fill up what is present in common? Isn't it? That is how we are using the overlapping DNA fragments to complete our DNA sequencing method. Okay, so that is the idea about what is Maxam-Gilbert chemical sequencing procedure. Now let's read this. So Alan Maxam and Walter Gilbert published a DNA sequencing method in 1977 based on chemical modification of the DNA and subsequent cleavage at specific bases. Okay, this method, unlike Sanger's DNA sequencing, lacked the use of primer I have discussed with you. So let's come to the steps. So first step is the radioactive labeling. Here I have prepared the note where we are adding the radioactive labeling when the DNA is in its denature double-stranded form. Okay, you can also use what I have discussed with you diagrammatically. Okay, both the process is same. Now we will cut the DNA fragment with specific restriction endonuclease, although for determining what is present at the 5' end, I will prefer you to use a restriction endonuclease mixture, okay, resulting in two unequal DNA fragment. Then we will simply denature it to convert them single stranded by increasing temperature, and cleavage the DNA strand at specific position by using chemical reaction. Okay, so here dimethyl sulfate and piperidine that we have discussed selectively attacks purine, and pyrimidine is attacked by hydrazine. Okay, and this process is called chemical modification, and this chemical treatment will generate break at the 4 nucleotide bases in the 4 reaction mixtures. So in the reaction mixture G, we have prepared our reaction mixture. So after reagent G condition, in the second test tube we will use reagent A plus G, that is designed in such a way that it will cut the DNA chain or break the DNA chain after adenine and guanine by simply modifying adenine and guanine. And in reagent C, it will break the DNA chain after the cytosine bases, and in the reagent C plus T, it will break the DNA chain after the cytosine and thymine. So as I have discussed with you that the concentration of the modifying chemical is controlled to introduce on an average 1 modification per DNA molecule. Thus a series of radioactively labeled fragment is generated starting from the radio labeled end to the first cut site on each molecule, that is after the base that we have modified with the use of chemical reagent. As a result, we have several differently sized DNA strands in 4 reaction tube. Fragments are subjected to electrophoresis, and here obviously page will be used, as page will provide us more higher resolution separation rather than agarose. Then we will visualize them with the help of autoradiograph if we are using radioactively labeled DNA fragment. Then the DNA sequencing procedure that we have discussed, that the gel will be read from the bottom to the top. The gel has nucleotide sequence differing by only one nucleotide. That means the subsequent base will behave only one nucleotide longer than the previous one. The larger the fragment, the more it is slowed down by the gel, and each band on the gel will indicate a specific nucleotide. Thus, by reading from the bottom of the top of the gel, the entire DNA fragment can be sequenced as the nucleotide indicated by the smallest fragment is closer to the 5' end. That I have discussed with you, and the nucleotide indicated by the larger fragment is closer to the 3' end. Okay. So that is how the entire DNA sequencing procedure in case of Maxam-Gilbert chemical sequencing procedure is done. Although the entire process is quite same with our Sanger's one in case of the red marked part, but we have to, the difference is here. Here we are using chemicals, and in case of Sanger's we are using dideoxynucleotides. That is the difference.